In 1779, just a few years after his colleague Joseph Priestley had discovered oxygen and noticed the creation and absorption of gases by plants, Jan Ingenhouse, while working in the same laboratory, placed plants underwater and using a transparent container, he noticed the leaves made bubbles when sunlight from a nearby window shined on them. When placed in darkness, however, he noticed that the plants mysteriously no longer seemed to emit the unknown gas. Holding a lit candle and exposing it to the unknown gas he had previously collected, he watched in amazement as the candle would burst into a flame. Ingenhouse would go on to make one of the greatest discoveries in all of science, that every plant, every leaf, and even a single blade of grass possessed the same incredible power. The power to produce the life force behind every living organism on Earth: oxygen. Though some other scientists would later attempt to claim the discovery for themselves, and Ingenhouse's work would not be recognized in his lifetime, it would be over another 100 years until the term referring to one of the greatest discoveries in science became known as photosynthesis. In photosynthesis, plants take water and carbon dioxide and use them to build sugar, releasing oxygen in the process. Photosynthesis is powered by light. If you look inside the leaves of every tree, every plant, and even every blade of grass, the energy and light is captured by the same fundamental process: the electron transport chain. The electron transport chain captures the energy and light and uses it to power several large molecular machines that are found in membranes in plant chloroplasts. The first machine is Photosystem II, which includes chlorophyll molecules that give plants their bright green color. Photosystem II splits water molecules into its component parts and uses them in different ways. A hydrogen ion is released at the top and its electron binds to chlorophyll. Chlorophyll absorbs light and energizes the electron. Photosystem II then transports the energized electron to the other side of the membrane, where they are placed on a small carrier molecule called plastoquinone. Then, to neutralize the negative charge of the electron, plastoquinone picks up free hydrogen ions from the surrounding environment. The electrons are transported to the next molecular machine called cytochrome B6F. Plastoquinone binds to the upper portion of cytochrome B6F, releasing its hydrogen ions. Notice that this has the overall effect of pumping hydrogen ions across the membrane. They are picked up at the bottom at the photosystem and released at the top at cytochrome B6F. Photosystem II splits another water molecule, continuing to release hydrogen ions and transport their electrons to plastoquinone. Finally, the oxygen atoms are combined and released as oxygen gas, and the whole cycle begins again. The electrons are transported through cytochrome B6F in a tricky way that pumps even more hydrogen ions. One electron passes up and is placed on a small carrier protein called plastocyanin. The other moves downwards through cytochrome B6F and is placed on another plastoquinone molecule. When this lower plastoquinone has gathered 2 electrons and 2 hydrogen ions, it flips to the top site, releasing its electrons and hydrogen ions in the same way. Notice this small cycle also has the effect of transporting hydrogen ions from the bottom side of the membrane to the top. The third set of proteins deliver the electrons to their final destination, a carrier molecule called NADP. A small soluble protein called plastocyanin delivers the electrons one at a time to photosystem 1. The electrons have lost energy at each step of their journey, so they are re-energized by light by the chlorophyll in photosystem 1. The energized electrons make a short journey from ferredoxin to ferredoxin reductase, which adds the electron to NADP along with a hydrogen ion taken from the surrounding environment. The electrons and hydrogen ions carried by NADP are then used throughout the cell to assist with the biosynthesis of sugar and other photosynthetic processes. During electron transport, many hydrogen ions are transported across the membrane, creating an electrochemical gradient that may be harnessed to generate energy. ATP synthase uses this gradient to connect ADP and phosphate, creating ATP. Hydrogen ions flow through ATP synthase, turning a rotary motor that powers the construction of ATP. This ATP is used to power most of the processes of the cell, including the construction of sugar from carbon dioxide and water. In this way, the molecular machines of the photosynthetic electron transport chain work together to capture the energy of light, using it to build energetic molecules that support photosynthesis and other needs of the cell.